Okay, students, let's go back through the serine protease discussion. So chymotrypsin is a protein that is a serine protease, and this is a great and well-studied example of catalytic activity. So let's go back through it. We have our chymotrypsin secreted in the pancreas, and what it's going to do is its substrate will be amino acid chains and that of ingested protein. So when we look at this particular picture, this is showing a lot of beta-pleated sheets, a little bit of alpha helical structures, and within the center part here, that's our active site. And the important amino acid residues in the active site are aspartate 102, histidine 57, and serine 195. So those are our three key players within the active site. So this yellow part here, that's our enzyme without all the rest of it. So the rest of it's there, but we're not focusing on it. And then it has the three important amino acids, the aspartate 102, histidine 57, and serine 195. What I'd like you to notice is that from our previous discussions in the last chapter, that particular nitrogen on histidine 57 is deprotonated because its pKr is 6.0. So it's deprotonated, so there are the lone pairs. All right, what you're seeing here is within the active site, there's some hydrogen bonding going on, and that helps to stabilize those residues and form a nice little pocket for our substrate. Okay, so let's take a look at our next one. Okay, so here's the mechanism of peptide hydrolysis. So here, I'm going to circle it here. This is the peptide that's been ingested, made its way through the stomach, now is in the intestine. And the whole point of this mechanism will be to cleave it into two separate chains. So the whole point is going to be that that peptide bond right there gets cleaved. So we'll have one chain, right, that gets cleaved into one chain and a second chain. Okay. All right, so let's start. Now, this is the overview of the process. And one of the important parts here is that it doesn't happen in one step, right? Very systematic processes. And what we want to focus on are the ideas that we've been learning in the previous slides regarding the enzyme kinetics, what is the role of the enzyme, and what is the chemistry behind the catalytic process. So we're going to take this in steps. Okay, so here's the first step. Here's our peptide chain that we want to split apart. Okay, one of the things we'll see later is that chymotrypsin is specific for cleaving, where the R group are aromatic, so it's going to preferentially prefer boy, that wasn't a good. Hold up, let me get my little eraser. So it's going to preferentially cleave there. So ones like, let's say, phenylalanine, for example. Okay, so in step one, here's our active site, and see this nitrogen with the lone pairs. It's going to grab the H from the serine. And then at the same time, so that's going to be acid-base reaction right there. At the same time, the serine has those lone pairs, and it's going to attack the carbonyl group. Now that carbon from organic chem, if you recall, has a partially positive carbon. And then if you think about it, those lone pairs can get kicked up there. So what we're seeing here is a concerted reaction where you have acid base, the nitrogen pulls the H off, and the oxygen attacks the carbonyl group. So that's what we're going to call there. So that is going to do what's called a nucleophilic attack. Okay, and a couple other things before we move on from the slide is that at this point, the substrate, right, initially comes in. This part here would be E plus S, forming the ES complex. They're attracted to each other through intermolecular forces. Binding energies released. And then the chemistry starts. So what's going to happen here is we start to see that we're now looking at the catalytic process, what chemical changes are going on. Okay, so let's look at that. So here, this was same as the previous, right? You have the nitrogen lone pair grabbing the H, so that's your acid base. That's a repeat from the previous slide. Here you have nucleophilic attack. And then what you'll notice there then is that we end up with this very interesting species. That's our tetrahedral intermediate. That's unstable. So if you remember in our energy diagram, check back to the previous lecture, you start off with your enzyme and substrate, the energy drops, and then it goes up to an intermediate, right? So we're up here in this energy. Okay, something has to stabilize this. So this yellow part is another part of the enzyme that helps stabilize this negative charge here, positively charged residue there. And then one of the interesting things here is that you actually have a covalent bond, right, between the enzyme and the substrate. Okay, so they're attached to each other. And if you think about it, one of the interesting things, I don't want to call it ES though, because that's going to be confusing. So I'm going to erase that. You have a covalent bond form. You're at the top of this energy diagram. And that's the whole role of the enzyme. This is where we're seeing a alternative pathway that would be difficult to do experimentally in a lab. You're seeing that even though the energy is high because it's unstable, it's still lower than an activation energy would have been without the enzyme. And you're really looking at the role of the enzyme in keeping the substrate there from diffusing out of the active site. This is showing the anion hole, how you're going to have some stabilization of that tetrahedral intermediate. And so if you're looking here, this has a negative charge. That's the CO that's now negatively charged up at the top. And it has to be stabilized through a positively charged residue on the enzyme, and that helps to keep it in place. Okay, so now at this point, our tetrahedral intermediate's unstable, right? So that's unstable. And what you're seeing between here and here is that the bond, the peptide bond, is broken. Okay, and the hydrogen that was on here, notice the one in blue, is now transferred to the to complete the amino terminus. So now you're going to have NH3 plus amino terminus of that polypeptide. Okay, so remember that's the polypeptide chain from the ingested protein, or breaking them down in the intestine. Okay, and so now that's this group here that's going to now diffuse out of the active site. The other half of the polypeptide is still attached to the enzyme. Okay. Now, one of the things I mentioned to you earlier was that chymotrypsin is very specific for peptide chains that have a large hydrophobic group as their R groups. And that's because within the chymotrypsin active site, there is a large deep hydrophobic pocket where the hydrophobic group on the peptide that's being cleaved can fit. So if the R group is like phenylalanine, tryptophan, then there's a deep hydrophobic pocket, which means that that particular pocket has hydrophobic residues that complement that. Okay, so here we go. We were here. The half of the polypeptide chain diffuses out. Okay, and then the rest of it is still attached. Oops, still attached to the enzyme. Okay, and now what you want to think of is that if my peptide chain split and now I have an NH3 plus that's now gone out, that I have to regenerate on the other side. I have to split it. I need the carboxylate end. If I look here, this O is not available because that's the oxygen on the serine, right? That's from the serine. So we have to have a source of oxygen to come in here, and a great source of it will be from the O from water. So water is going to come in, and it's going to do a nucleophilic attack on the carbonyl group. So it has its lone pairs. It's going to attack it. Okay, and we're going to form once again another tetrahedral intermediate. Okay, so we have another spot where you have this kind of awkward looking structure where it's still attached to the enzyme. Okay, and so this was from the previous slide, so we're going this direction to this one. And we have then another tetrahedral intermediate because the water did nucleophilic attack. The tetrahedral intermediate is unstable. And so what will happen is that this bond between the enzyme and the substrate will now break. And then you have some cleanup here where you have to regenerate the serine. So it's going to grab a hydrogen. So the serine residue is regenerated. Okay, and now you have the other half of the polypeptide chain. Okay, so here is the regeneration of the enzyme. So you're back. This now is the other half of the polypeptide chain here. It's going to be released, and then you have your enzyme that's ready again. So now it's ready to start that whole process again. So I'm going to get to this slide here. Those three previous ones, you can read them, but one of them is just a rehash of it in a different image. But the important pieces to understand is that in catalysis, there's chemistry going on. And in this case, with the serine protease, what we're really observing is acid-base catalysis and nucleophilic substitution, or what we were calling nucleophilic attack. Okay, and the nucleophiles were the serine and the water in the later steps. And then for acid-base catalysis, we're looking at histidine and serine. Okay, and this is really hopefully you're seeing that when we look at these enzymes, they're just not these circular blobs that we just look at, that there's chemistry going on in that active site.